Hi, I'm Raj Giridharagopal. I'm a research scientist at the University of Washington, and I'm going to present our recent work in developing a method called non-stationary Fourier mode decomposition, which is a time frequency analysis method that we're applying to atomic force microscopy, specifically in the context of time-resolved electrostatic force data. Now, the motivation for this work is that there's kind of this growing trend in scanning probe microscopy over roughly the past decade and a half or so of doing big data scanning probe analysis. That is, when you're acquiring your data at each pixel instead of just getting X and Y and some measurement like the height or the current, you're also recording the entire cantilever signal versus time, taking all that data and then post-processing it. So this exploits the fact that digitizing data and data storage are both uh, easy and relatively inexpensive these days. So you can take all your data, analyze it after the fact, and try and extract some new information out of it. And so the result is when you have these large multi-dimensional data cubes that have all this information, you can take that data and try and learn some new dynamics. Now, the reason that we're interested in dynamics, of course, is that these dynamic processes are ubiquitous in materials. This ranges from things like carrier dynamics in semiconductors. Hopefully, you saw this movie. This is photovoltage dynamics in a tungsten diselenide 2D material, or photocharging in an organic solar cell. So this is showing the cantilever frequency over time, or ion motion in a polymer. And there's many more cases where we see dynamic processes that are both ubiquitous and nanostructured in nature. So we want to extract them with AFM. Now, the way that we do that is using a method called time-resolved electrostatic force microscopy. So what this method does is we apply an excitation, record the cantilever data, and then convert that cantilever data into frequency versus time. So let's go into that a little bit more detail because that motivates why we develop this new method for extracting that data. So first we apply an excitation. So that might be, say, a pulsed LED on a solar cell. And then in our AFM, we're recording that response. So what's happening when we apply this excitation is, as long as we have a voltage between the tip and the substrate, is we're causing a change in the resonance frequency. So that kind of obeys this general equation here, which if you've read any AFM papers about even Kelvin probe or EFM, should look familiar. But this delta omega is the resonance frequency of the cantilever, and the term that's circled is the important part. So that's the differential capacitance gradient or the electrostatic force gradient. And so what's happening is when you apply a pulse, like say an LED that causes charge to build up in a solar cell, this term changes in a time-dependent fashion. And I should pause here to note that the reason that the title of this talk includes non-stationary is when you have a signal that has a kind of time-dependent change in frequency that's not very well defined, that is kind of by definition a non-stationary signal, to use signal processing field's parlance. So that's what's happening in the AFM cantilever. We apply a pulse, we see a time-dependent change in the resonance frequency. Now, in order to measure that, we have to record that data. So this is an example of the recording where the y-axis is the deflection signal that you might get from the photodiode, and the x-axis is time. So the cantilever is behaving normally, then we apply an excitation, then the cantilever's amplitude will change because its resonance frequency is changing. And if we zoom into this green blob here, it looks like just solid circle or a solid colored graph, but if you zoom in, there's actually a sine wave that corresponds to the cantilever's deflection. It's oscillation going up and down, up and down. So that means we take our raw cantilever signal, or we take our cantilever, we've applied an excitation to it, and we record it. The final step, once we have our raw cantilever signal, is we demodulate it to get the instantaneous frequency. So this could be done using a number of different ways. I think MATLAB even has a built-in function called demod. We can use like the Hilbert transform or short-time Fourier transform or wavelet transforms, anything that converts the signal versus time into a frequency versus time. So that gets this graph like as shown in the middle here, where the frequency is on the y-axis, time is on the x-axis, and this corresponds exactly with the time of our signal up here. So the cantilever, there's no excitation, so it hasn't shifted yet. Then once you apply an excitation, the cantilever's frequency will shift away. So in this case, minus 1200 hertz, and then eventually it'll relax back to its original position. And then at the same time that we're doing this demodulation, you can also extract the amplitude, which is effectively the envelope of this green function at the top. So that's how time-resolved EFM works. Now, there are some natural limitations to this technique, which is sort of the motivation behind we wanted to develop a new method. There's actually two limitations. The first is that we kind of are not extracting the information that we want. So in the case of, let's say, photocharging in a solar cell, you apply a light, charge builds up in the solar cell and is changing your resonance frequency. So that happens in kind of a single exponential fashion. A lot of these kinetics are sort of single exponential by nature. So it builds up some amount of charge and then it naturally saturates. So this might be the behavior that we expect in blue. So a single exponential curve. The thing that you measure though is like these instantaneous frequency curves are in red. So you see this dip and then it eventually relaxes back. So that means what we want to measure the blue, but what we actually measure is the red. So you can kind of get around that. And the way we do that right now is you build up a calibration curve by taking your same cantilever, putting it on a gold substrate and applying fixed exponential pulses of a well-defined character out of a function generator, then generating this giant calibration curve, and then taking your actual experimental data and then fitting them to a function that's a spline of this curve. That's kind of tedious. Your tip has to also survive the entire process in order for you to do it. And it's kind of a roundabout way, because that's just saying we have this red curve, we apply this function here, and then eventually we get these blue data out. What we want to do is just directly get this blue data. We want to just get the transient directly. So that's one limitation. The other limitation is related to noise. Demodulation methods like the Hilbert transform or short time Fourier transform or continuous wavelet transform, et cetera, they're kind of naive. They don't adapt. You just put your data in, you get your data out. So that can be kind of a noisy process. So in this case, here's our organic solar cell. 2 different points. We have a slow pixel in red and a fast pixel in blue. This is what we get out of the Hilbert transform. And I mean, you can see the trend there, but it is, it's reasonably noisy. But if we have like a more difficult signal, like this toy signal down here, the result is actually you can get very noisy results. So what we'd like to do is a method that can get our hidden information and do it in a higher signal to noise fashion. And the way that we do that is non-stationary Fourier mode decomposition. This builds upon actually something called Fourier mode decomposition, which is also by Nathan Kutz's group that's published this year in this reference here, where that uses an iterative method of stochastic gradient descent to learn the correct frequency of a signal. In this case, we have our initial guess in orange and then eventually learns the correct signal. NFMD is taking this approach, but applying it to windows of the data as we slide along our signal. So at a particular window like this outlined in this yellow box, here's our raw signal. We then apply NFMD to learn the correct frequency and decompose it into different modes. We say that our signal is composed of a sum of different modes of the signal, like the 0th mode might be the mean, and then the first mode might be the actual frequency, et cetera. So we get a time-dependent frequency and time-dependent actually set of frequency modes for our data from this mode decomposition by sliding this window across. And for those of you that are interested in some of the nuts and bolts, you can look at actually some of the data. The original code is in Dan's GitHub repository down there, and we specifically use PyTorch in order to do this. So that's how this NFMD works. You slide this window across, learn the correct frequency via stochastic gradient descent, move to the next window, and then repeat. It has a lot of key advantages. The first is that you get much higher signal to noise ratio. Like in our toy signal here, this is our original Hilbert transform data. But you can see in the Fourier mode decomposition, it's already just very obvious by eye that it's much lower noise on that signal. Another key advantage of NFMD is it responds very easily because to instantaneous jumps in the frequency, like might happen if you apply, say, a sharp voltage step or a huge light pulse to a solar cell. So that's on this graph at the bottom here, where the black is our ground truth and frequency, where there's a sharp change in the frequency in black. NFMD in orange will actually track that pretty nicely. And you can see it barely deviates from the ground truth, whereas like the Hilbert transform deviates a lot. That's in blue. Now, the way you get around that from like the Hilbert transform approach is you just take a lot of data and average it together, and eventually you get a high enough signal to noise that it doesn't matter. But NFMD can do that at much lower noise threshold. And it's actually pretty obvious when we look at experimental data. So here's our good organic solar cell data again. Here's the Hilbert transform data that I was showing a couple of slides ago from these same 2 pixels on the image. And if we apply NFMD to the same set of data, you can see already just visually that it's much cleaner. It's much lower signal to, or much higher signal to noise. This is for the same window size as sort of corresponds to what we get out of the Hilbert transform here. So that means for like the same conditions, we can get much higher signal to noise from NFMD. Another particular advantage, though, which is not just in cases where the Hilbert transform works, but it's nice in cases where the Hilbert transform doesn't work. So like in this case, this is on a perovskite solar cell data. And incidentally, this is data I took at Oak Ridge with Liam Collins. Here's our Hilbert transform data on the left, and you could basically not see anything. And then for NFMD of the same data for kind of the same conditions, we can actually extract out the two, the slower and the faster pixel in the blue and the red respectively, as shown at the top. Now it's noisy, so if we increase the window size or take more averages, et cetera, then we'll clean up the noise. And in fact, for the Hilbert transform data from this paper, what we did is we took lots and lots of data and averaged it together to clean up the signal to noise in order to extract and get a really nice image down here. But this is showing that with a much lower number of data points, a number of signals, we can get the same basic data out of NFMD. So that means you can get data, you get higher signal to noise in general, and you require a fewer actual number of acquisitions in order to get a reasonable signal out of NFMD. So that solves problem number one. The other thing that I pointed out, the limitation of current techniques, is that when we're feeding in something, or we want to see single exponential kinetics or some sort of reasonable kinetics, and when we get at these kind of dips in this relaxation back in instantaneous frequency, and that's just a nature of what happens to a cantilever if you solve the differential equation for how a cantilever responds in response to a time-dependent change in its resonance frequency. That's just going to happen. So how do we get that out? So NFMD can kind of extract that in a pretty cool way. So Dan Shea, who wrote this, discovered this kind of interesting technique. So in this experiment that we have on the left, these are simulations of applying a voltage pulse with a fixed exponential time character to a substrate. So that's in black. And so these are for different time constants that we're applying. So for a much slower, this is like 1 millisecond and eventually 100 microseconds, et cetera, to the substrate. And then seeing, this is fitting the mean mode of the NFMD response. So kind of like the 0th order mode that we're getting out of this Fourier mode decomposition and feeding that over time. So the x-axis is time, the y-axis is kind of the mean position. And what this shows is that kind of the average mean, like the time-dependent mean of the signal, actually tracks this excitation response pretty nicely. So that's the black is the ground truth. The colored curves correspond to the time-dependent mean. And you can see this tracks actually really nicely up until around the red curve, which corresponds to 1 microsecond. And that's kind of well defined at this graph at the right. So on the x-axis, you have our true time constant that we're feeding into the simulation for the excitation that we're applying. The y-axis is the predicted time constant by trying to fit to this time-dependent mean. And you see these points correspond or line up really nicely up until around 1 microsecond, which is 1/3 of a cantilever cycle. So we can get sub-cycle directly probing the time constant. And again, in this case, we're directly measuring the single exponential response that we wanted out. So we feed in a single exponential, we use NFMD, we get that single exponential data out. So that's really cool. This solves kind of a longstanding issue that we've had in these time-dependent AFM measurements, that we're directly probing the dynamics. So in conclusion, this NFMD method both gives us better signal-to-noise ratio, and it lets us extract this hidden information out from these time-dependent AFM experiments. And in the future, we want to apply these to generate better source data for, say, neural networks, as well as applying these on cases where you don't have single exponential, maybe you have stress exponential kinetics, and we can kind of use that knowledge to guide the NFMD process. So with that, I would like to acknowledge all the people. Dan Shea is the one who did all the coding and analysis, and I supplied all the experimental data, and as well as David Ginger, Steve Brunton, and Nathan Kutz, and our funding. And I'd like to note here, because I'm recording this in advance, that you should pause, go to the program, and find Madeline Brashear's poster presentation, also from the Ginger Lab, where she'll present some interesting work on neural network analysis of time-resolved EFM. So with that, thank you for listening.